Okay, so this is our video lecture on the central dogma of molecular biology. We are in the middle of the genetics unit. The objectives are listed here. Please make sure you read through them. Remember, when you get to the end of the video, it's a good idea to come back here and see if you can answer them with a thorough explanation. As always, if you have any questions, contact your teacher. Okay, let's start out by talking about DNA. This is a little bit of a review here. We've talked about DNA before. It stands for deoxyribonucleic acid. It is a polymer which is built of nucleotide monomers, and those nucleotides have a sugar, a phosphate, and a base. So if you see over here on this diagram, the P stands for the phosphate group, the S stands for the sugar, and then connected out off of this arm, this A is one of the possible bases. We know that there are 4 different bases that are in DNA. You can have A, you could have T, you could have G, or down here you could have C. Okay? And so that's what DNA is made up of, and it's this very interesting molecule. Watson and Crick have been given the Nobel Prize in 1953 for coming up with the structure of it, but they could not have done it without reading a paper written by Erwin Chargaff. And so Chargaff's rules give us the base pairing rule, which basically says in the DNA molecule, A and T will always bond together with one another, and G and C will always bond together with one another. So when you look across the DNA molecule and you see it drawn like this, it always looks like there's sort of 2 little sticks connecting together. What that's representing is the bond between the base pairs. And something of interesting note, if you notice that we are using hydrogen bonds to hold these together, between the A and the T we have 2 hydrogen bonds, but between the C and the G we have 3 hydrogen bonds. So the different shapes of the structures of those nucleotides, of those nitrogen bases, determines how many hydrogen bonds are going to form between them. And last thing we know, DNA is always contained in the nucleus. The one exception is during interphase, when we are replicating our DNA. So this whole replication and then reading the DNA to make protein is known as the central dogma of molecular biology. And basically, a dogma is sort of like a strong system of beliefs. You may have heard of it in religion if you're familiar with Catholicism. And this is a strong, core central theme of beliefs that we have as far as our understanding of biology. This diagram here sums it up really nicely. We have DNA replication that's happening over and over again. And what that does is that allows our information to be passed from cell to cell and from generation to generation. But then what do we do with it once it's inside a cell and we're not worried about passing it on? Then that DNA is going to be transcribed through the transcription process, and then we have translation that occurs. So in the transcription process, what we're going to see is that DNA is going to be rewritten into a new molecule known as RNA. We're going to talk about that on the next slide. And then that RNA molecule will be translated into protein. So there's sort of this flow of information as we replicate our DNA, and then we transcribe and translate into our end goal, which is protein. So we take our DNA molecule. Let's just focus on the replication portion right now. We know that this occurs during interphase of either mitosis or meiosis. And this is going to be when we take one DNA molecule and we make 2 DNA molecules so they can go into the 2 resulting cells, like in mitosis. Well, we can also use Chargaff's rules to help explain how DNA copies itself. So when you look at the DNA molecule, and you see those bases, how they are bonded together, A always pairs with T, and C always pairs with G. We're going to split this DNA molecule right down the middle. So we're going to separate each strand from one another. And that enzyme that does that splitting is called helicase. Okay? So now here you see a section of DNA that is still together, and then the helicase molecule, which is represented by that gray rectangle there, that is showing what's going to happen. Helicase is sort of like a little special scissors. They break those hydrogen bonds that are holding the base pairs together and allow them to separate from one another. Okay, let's focus in on this diagram then for the next part. So in the nucleus, okay, floating around in the cytoplasm that is in there, there are a whole bunch of free nucleotides, just random A's, T's, C's, and G's kind of hanging out, waiting for replication to occur. So once this molecule is split open and you have one strand here and one strand here, you have these lonely little bases. This A says, hey, I'm missing my T. And this C says, hey, I'm missing my G. And so what's going to happen is another enzyme is going to come along, and that's going to be called DNA polymerase. And what DNA polymerase will do is it's going to join together the free nucleotides that are in the cytoplasm, and it is going to base pair them with each side of the strand. So you'll see the blue represents the original piece of the DNA molecule. The red represents the new sequence of nucleotides that the polymerase is building. So if we start down here with polymerase and it says, okay, it's going to read an A on the original strand, which means it's going to need to build a T on the new strand. So it pulls a T out of the cytoplasm and attaches it to the A. So now you have a base pair and that bond forms. Then it moves on. I have an A, I need another T. I have a G, I need another C. I have a C, I need another G. Okay? And that's how it's going to synthesize the strand. Now while that's happening on this side of the molecule, it's also happening over here at the same time. So we have multiple polymerase molecules working to build new complementary strands to each of the resulting DNA molecules. The other cool thing that polymerase does is it's able to proofread itself. So, it goes on for, let's say, 1,000 nucleotides. Once it finishes, it goes back and it reads everything to make sure, yep, A with T, A with T, T with A, C with G, G. And if it comes to like A with C, it says, oh, we have a problem here. And then that's when one of the other enzymes in the cell is able to fix it. So DNA polymerase not only builds the new strand in each of the resulting molecules, but it also proofreads itself in order to reduce the chance of error and therefore mutations. This form of replication is known as semi-conservative because we know we get two identical DNA molecules here, but they are semi-conservative in the sense that semi means half and conserve means to save. If you look at the coloring here, you see the parent DNA is represented by yellow. Well, when that split happens and we build two new identical DNA molecules, half of each molecule is still from the original parent DNA. So that's why you see one half is yellow and the other half is red, and the red represents the newly synthesized strand. So each of the two resulting DNA molecules are going to have one original strand and one new strand. So now we know how we're able to pass on that information from cell to cell and generation to generation. So let's read that DNA and use it for what it's needed for, and that is protein synthesis. So we're going to go through two processes here. The first one is called transcription, and the second one is called translation. And before we get into the details of each of them, let's just use an analogy so you understand what's happening at each step. Okay? In transcription, we're going to take our DNA, which is here, and we're going to rewrite it. That's what transcribe means. Okay? We're going to rewrite it into an RNA molecule. Specifically, it's called mRNA, and I'll explain that in a future slide. Then we take that mRNA, we're going to ship it out of the nucleus, and we're going to send it to a ribosome, and the ribosome is going to read the information on that RNA, and it's going to use that information, it's going to decode it to determine what amino acids to link together to build a polypeptide. And as we know, a polypeptide is the polymer of a protein. So this explains how we build protein. So let's start with an analogy. Okay, one of my favorite things, I love food, right? And my best friend from college, Jewel, she makes the best banana bread I know. She has her own special recipe for it. She has, modified it over and over. And I want to make some banana bread. So what do? Okay, first step is I need to get a hold of her cookbook. Okay, and her cookbook is like the DNA molecule in the sense that it contains all of the recipes that she uses in order to produce all of the food products that she wants to make. There's recipes for soups, there's salads, there's appetizers, there's desserts, there's all sorts of stuff. And of course, there's banana bread. So it's like your DNA molecule in the sense that your DNA contains genes, which are the recipes for making all of the proteins that are going to be produced in your body. But I don't want the whole cookbook. I just want the banana bread recipe. So what I'm going to do is I'm going to flip open the cookbook to the banana bread page, and then I'm going to make a copy of it. And I'm going to copy that onto a recipe card. So I'm physically going to take a pencil and a paper, and I'm going to write out the recipe for the banana bread, 4 slightly ripened bananas, 3 cups of flour, 2 cups of sugar, whatever it might be. This is like RNA because the RNA is a single copy of a single gene. So just one recipe that we're interested in to make one protein. Then I'm going to decode the information in that recipe. I'm going to read 3 cups of flour, and I'm going to get my ingredients together, and I'm going to mix them together appropriately according to the instructions, and voila, at the end I get yummy banana bread. Walnuts, it depends on the day. Sometimes yes, sometimes no. Okay, so keep this analogy in the back of your head that we have the original cookbook. We're going to make a copy of one recipe, and we're going to copy that down. Then we're going to take that copy and we're going to translate it. We're going to read it and decode what each of the pieces mean, and we're going to use that to build our ultimate end product. So we go from DNA, which we're very familiar with. Next step is we make RNA. Well, what the heck is RNA? So focusing on DNA molecules, we know that they have 2 sides. They use a deoxyribose as their sugar molecule. Remember, the nucleotide is made up of a sugar, a phosphate, and a base. So this sugar just has a special name. The nucleotides in DNA, we've got A, T, C, and G. And DNA functions as the blueprint for how to make every protein in your body. So it's the whole cookbook. Okay? So what's this RNA business? Well, there's actually lots of different types of RNA molecules, but we're just going to focus on one type in this lecture. So RNA molecules, they're different because they are single-stranded instead of double-stranded, and they use ribose as their sugar, not deoxyribose. So a slight change in the structure of the sugar, not very significant. One thing that is really significant is when we look at the nucleotides, in DNA we had ATCG, but here, instead of thymine, we find that RNA uses U, which is we're naming uracil. And while thymine and uracil are very similar in structure, they're different. And I always get students who ask, well, why? Why does RNA have uracil and not thymine? Sometimes in science, the answer is this is what we have discovered. We can't explain the reasoning behind it, but this is just how it is. So when they analyzed pieces of RNA, they found that this is a different chemical base that was being used, not thymine, it was uracil. So sometimes you just have to say, this is what we have discovered. There's no explanation behind it. And this RNA molecule, just like our recipe card, it's a disposable copy of a single gene in order to make a single protein. All right, and that is the role of mRNA, and we'll talk about mRNA in a second. So let's go with step 1. Of the first 2 T's, we've got transcription and then translation. So starting with transcription. When you think of the word transcription, you want to think of the word to transcribe, and that means to rewrite. Okay, so in in your class, you may be asked to rewrite your notes, or you might rewrite your notes when you are studying for a test or something like that. So when you are transcribing something during protein synthesis, you're going to rewrite a section of DNA into a sequence of mRNA. Remember, the RNA molecule, single-stranded disposable copy of just one gene from the DNA. This is happening all in the nucleus. So we call this mRNA M to specify what type it is. The M stands for messenger. Okay? And when you think about what does a messenger do, it picks up something, takes it to where it needs to go, and drops it off. And that's the same idea here. The mRNA molecule is going to make a copy of that one gene from the DNA, and then is going to transport that message out of the nucleus where it's going to be translated in the ribosome. Okay? So here you see a nice diagram here. We can zoom in on this part. All right? You'll see that we have the blue and the red both representing our DNA molecule because it's double-stranded here. We split apart the DNA molecule using an enzyme, and then what's going to happen is instead of DNA polymerase, RNA polymerase is going to start grabbing RNA nucleotides and start building a sequence of mRNA. So you'll see the C goes with the G, T goes with the A, but here's a big change, okay? Instead of A with T on the RNA strand, A is now going to bond with U, and so it's going to call for the uracil nucleotide as it builds this growing mRNA sequence, right? And it's going to continue until the gene sequence is done. So things to remember, you just want to remember A to, I'm sorry, jeez, scratch that out. We're going to go T to A. We're going to go C to G. We're going to go G to C. And then most importantly, A to U. Okay, so DNA is on this side and the RNA complementary nucleotide is on this side. So A to U is the big difference here. Alright, so it's your turn. Here's a sequence of DNA. What I want you to do is I want you to transcribe it and tell me what the mRNA sequence would be. So go ahead and pause the video, and when you're done, alright, so here is the complementary sequence that you should have come up with. Double check it to make sure that you followed your proper rules, noticing here that we have, noticing here that we have A, and then that is going to bond with U is your mRNA code here. Okay, so you see we have this sequence of mRNA, and now it is ready. Okay, this is the complete sequence. This is my recipe for banana bread. I rewrote it. Now I can travel to the ribosome, and that's where translation is going to occur. Part 2 is called translation, and when you think of translation, you should think about languages, right? When you take Spanish, you have to translate from one language, into another. So if I say, house, I have to be able to translate that into casa in Spanish, okay? Or whatever word or whatever language you can think about. But there has to be some sort of a code that's going on here. So there's a conversion that's occurring. So in translation, we're going to take the order of those mRNA nucleotides, and we're going to find out that they contain a secret code that determines the sequence of amino acids that we're going to use to build our protein. Again, this is happening in the ribosome. That's why way back when in chapter 3 we said that the ribosome is the site of protein synthesis. One thing that scientists have discovered through many, many long experiments is that when you read the mRNA letters, okay, when you read the sequence of the nucleotides, they're grouped together in threes. Okay, you read 3 at a time, and that's called a codon. And each codon codes for one specific amino acid. Okay, so if you have the sequence GAC, that would code for a specific amino acid, right? Think of the amino acids as the ingredients that are going to be used to build my banana bread. So my individual flour, and my sugar, and my eggs, and my butter, and all those things are going to come together in a specific order, right? Remember the dry to wet versus wet to dry, and other things like that about a recipe. That's going to determine what amino acid is going to go in the chain of the polypeptide. Right? So here's what I'm talking about. So down here on the bottom, we've got the black line here, and that represents our mRNA. So let me just get some here. This is our mRNA sequence, okay? And it is being read through the ribosome, and the ribosome reads in groups of 3. So C, G, G. Those are the 3 nucleotides that get read together. That's a codon, and that codon is going to code for an amino acid that is named arginine, okay? But this codon, CUU, that's going to code for an amino acid named leucine, and AAG codes for lysine, etc., etc. So this is going to keep happening. So the ribosome is reading the mRNA, letter by letter, and as it groups them together in 3s, then these amino acids, which are in our cytoplasm, are going to arrive at the ribosome. They're going to get transported there by another molecule. And they're going to arrive at the ribosome, and then they're going to start forming a chain. So the sequence is very important here. First arginine, then leucine, then lysine, then arginine again, and then tyrosine. So this sequence is going to be used to build a protein. When each of these amino acids arrive, they're single. But what's going to happen is as soon as they get next to one another, they're going to form a bond together, and that is called a peptide bond. Again, this is a review of what we talked about in our macromolecules lesson. So in order to know that CCG goes with proline, we have to have some sort of a code. All right? When we talk about language, there's a dictionary, right? An English to Spanish dictionary. It shows what word goes with what. We have what's called a genetic code. And so here's the way that you read this diagram. Notice that there are 3 rings of RNA nucleotides. They're sort of different sizes and different colors. Starting in the very center, when you read a codon, for example, this codon here is AGG. This is the first position, second position, and third position. Starting in the center with A, all right, what that does is that narrows down the possibility to just this corner. So we have A. So put your finger on A. We'll follow along here. So we've got A here. My second one is G. So connecting to A, I need to go to G. And then my third one is G as well, all the way out to this ring. And so I find that my answer is arginine. Okay? But let's say I was looking for, I don't know, let's do UCU. Okay? First letter is U, second letter is C, third letter is U, and so that tells me it would be serine. Okay? Try and practice, see if you can write up a couple of different RNA codons, and then see if you can figure out what amino acid they would be. Note that when you look at this genetic code, it tells you a few things. First of all, each amino acid isn't necessarily unique. For example, UCU, UCC, UCA, and UCG are all going to code for serine. So they're all going to code for the same amino acid. Some of them, however, are going to be just for one. For example, AUG will code for just one. So we have 64 different possible codons, okay? So 64 codons, and there are 20 different amino acids. All right, so that explains why more than one codon can code for the same amino acid. So let's take this all and put it together. We're going to be making a sequence. So how do we know where the sequence starts, and how do we know where the sequence ends? The start codon is AUG, and the way that I tell my kids to remember that, it's the only one you have to remember. AUG. When does school start? It starts in August. What's the abbreviation for August? AUG. Okay, and one interesting note here, all protein amino acid sequences are going to begin with this AUG codon, which means that all protein sequences start with the methionine. And so when you think about that, that's a really neat thing to think about as far as evolution goes. because that's not just for humans, that's not just for animals, that's for every living thing. Every living thing. It shows that unity, remember how we talked about the unity and diversity? For how different we all are, one thing that unites us is the way that we produce proteins, and this translation is going to occur in all cells, prokaryotic and eukaryotic, in the same way. So all sequences will always start with methionine, or I abbreviated it here, MET. And then all sequences, you need to know where to stop. And so there are 3 codons that scientists have found. These are going to determine the end of an amino acid sequence, and when the ribosome reads that, it releases that polypeptide chain from the ribosome and allows it to go off and do whatever it is, whether it's a polypeptide that's going to be twisted and folded into a transport protein and added to the cell membrane, or whether it's a polypeptide that's going to be twisted and folded into an enzyme that can bond to a specific substrate. Many different proteins, many different responsibilities. Alright, so it's your turn. So we've already done the transcription part. You went from DNA to mRNA. So here's that same mRNA sequence. What I want you to do is pause the video, use the genetic code that was back a few slides, and see if you can determine what the amino acid sequence is. And just a quick note here, you will not have to memorize that genetic code. You will always be given the genetic code. The only thing that you need to know is that that start codon is always where you begin translation. So that AUG, and it codes for methionine. So pause the video here. Alright, so let's see what you came up with. Hopefully you got this sequence. This is methionine, then we have alanine, then we have isoleucine, then we have arginine, then we have aspartic acid, and next we reached a stop codon. So we just put stop, or that lets us know here's the beginning, here's the ending. Okay? So one of your objectives, you need to be able to look at a sequence of DNA and then actually transcribe and translate it, just like we've been doing on these slides, which are titled It's Your Turn. Okay? So this is how every protein in your body is made. It goes through the transcription and the translation. And I just want to bring it back to chapter 3. We talked about the endomembrane system, looking at all these organelles that work together in order to do this process, right? Starting with the nucleus all the way at the top here, which is going to produce the mRNA. You can even see that the membrane that wraps around the nucleus is physically connected to the rough and the smooth ER. And remember, pick out one of these ribosomes, and let's say that's where that polypeptide is originally built. Its chain has been built there. Okay, and then it's going to move through the ER, and that's where that's going to receive all sorts of modifications. It's going to be twisted and folded into its final protein shape, moving through the smooth ER if necessary. We see it jumping from one organelle to the next in these transport vesicles. Here that vesicle is fusing with the Golgi apparatus, and the Golgi apparatus is going to package it into a secretory vesicle that we see over here, right? And that's, remember, the Golgi is like the UPS system. It puts those proteins into a secretory vesicle, and then that vesicle is going to fuse with the cell membrane, and those proteins are going to be exported. Same thing is going to happen as we take in proteins, and these proteins are whole, but we're going to use them as the building blocks to make new proteins. So it's sort of like if you have a pack of Legos. Okay, everyone had Legos as a kid, right? I think you did. I hope I'm not too old. You have a big bucket of Legos, and one day you build a ship. Okay? For that day, those Lego blocks are part of a ship. But as soon as you take that ship apart and you break it back down into its individual Lego building blocks, those building blocks can be used to make anything else. So tomorrow, you don't feel like building a ship. Instead, tomorrow, you're going to take the same Lego blocks and you're going to use it to build a house. Okay? Same idea. All of the proteins, when you think about the foods that you eat, you break down those proteins into their basic amino acids, okay, those individual building blocks, and then we use those to make new proteins that we need. For example, new amylase enzymes, or new hemoglobin molecules, which help to carry oxygen in your body, or all of the other, I don't know, hundreds of thousands of proteins in your body. So this endomembrane system plays a very important role. It shows a system, which is how these organelles are going to work together for one purpose. So they have many different structures, but they all have a related function. All right? And lastly, again, I just want to touch on the universality of how transcription and translation occurs in all cell types, prokaryotes and eukaryotes, though there are some slight differences. Okay, just looking at these diagrams here, we have a prokaryotic cell on the left and we have a eukaryotic cell on the right. The first thing that should look really different is the fact that the eukaryotic cell has a nucleus. And so because of that, transcription is going to occur separately. It's going to occur within the nucleus, and the mRNA strand must be made as a whole, all in one shot, before it moves out of the nucleus into the cytoplasm where the ribosomes are and where our amino acid chains can be built. But when you look at a prokaryotic cell, their DNA is just free floating in the cytoplasm. And so what that means is transcription can begin. And as that mRNA strand is snaking away from the DNA molecule, ribosomes can attach on and start translating it as well. So we can build our amino acid chain while the mRNA is still being transcribed. So it's sort of like it's happening all at the same time. But similarities, note that we still have DNA going to mRNA. We still have the ribosomes, because remember, ribosomes are the one organelle, the true organelle that prokaryotes and eukaryotes both have. So we have these ribosomes, which are absolutely essential to prokaryotic cells, because that's still where we're going to build the amino acid chains, which will ultimately fold and twist into our proteins. Okay? So, I know that was a lot, and I know this video got a little long, but please, if you have any questions, make sure that you email your teacher, and we will go over this in class. Thanks.